Hello, welcome to Exam2 Essay1. My name is Jacob Nelson, and here's my submission for Bio461's exam questions. Okay, describe the molecular characteristics of the contractile filaments found within the functional unit of the skeletal muscle cell. We have a neuron that makes a synapse with a muscle cell, and the muscle cell will react to the depolarization from the synapse via acetylcholine neurotransmitters entering the cell, and this allows receptors for potassium to exit the cell as sodium rushes into the cell, creating that synaptic propagation that goes down to what we call a T-tubule. The T-tubules contain other protein channels, and more specifically, one we call a DHP channel for calcium. When calcium reacts with this channel, it opens up a RYR channel that is connected to something called the sarcoplasmic reticulum. And this houses a bunch of calcium for the muscle cell. When the action propagation occurs, it reacts with this RYR channel to release calcium from the cytoplasmic reticulum into the cytoplasm of the cell. It doesn't leave the cell, it stays in it, but leaves the SR unit. As that happens, calcium is released into the cell, and it binds to different proteins, or it activates certain proteins in the muscle to make contraction. What it will do is bind to troponin, the type of protein that will connect to actin filaments and something we call tropomyosin. Now, these actin filaments have binding sites that the tropomyosin connects to, right? It is considered to happen when the muscle's in a relaxed state. But what troponin will do is come in once the calcium is released and binds to troponin. It'll go to these actin filaments and tropomyosin filaments. It'll conform its shape and move the tropomyosin away from the actin filament, and it will expose some binding sites. Now, these binding sites are going to have a high affinity with myosin. Myosin is another protein that is connected to thick filaments of the muscle, and once myosin is able to bind to those binding sites on actin, through the cycle that it has, it will then pull on these actin filaments. And as this happens, it happens throughout all the muscle fibers in a specific muscle, and it will actually shorten the filaments to come crossing together, and thus you have a muscle contraction. The mechanism by which an action potential in the sarcolemma results in release of calcium, I mentioned that. Like I said, it's once the cell is depolarized, action potential occurs, and this propagation of energy goes to these DHP ROI channels. And this will cause the calcium to release from the cytoplasmic reticulum into the cell, and these will bind into certain proteins to cause conformation changes. And then the sources of energy utilized in muscle contraction is a great one. So first source of energy that the muscle will typically use first is cytoplasmic ATP. So we mentioned myosin has this affinity to bind to the binding sites of actin. And how does it actually bind, and how does it pull on this actin filament? Well, it uses ATP. And the first source of ATP you have is in the cytoplasm. It's just residually there. There's a small amount, and will probably be used up within the first 1 to 5 seconds of the contraction. The second place you'll typically find a energy source of ATP. This will be in the what we call the M line of the muscle. It's just this imaginary line that contains, it's going down the middle of a thick filament, and where we will find different proteins like creatine and such. There is a ADP molecule found there, and this ADP molecule will bind with adenosine kinase that will then give us adenosine phosphate. You put those together, it just makes ATP. Now this will only allow you to have an additional 5 to maybe 10 seconds more of muscle contraction through ATP. So not a lot longer, but it is another source. Something else we have is what we call glycolysis. This is a biological mechanism by which the body intakes glucose and will create ATP through its glycolysis cycle, and eventually will be used up. I think this only will allow you to have 30 seconds to 30 minutes worth of ATP. So not very long, but it's a good amount of time. And then the final form of energy source we have is actually what separates this one from the previous three I mentioned. The previous three are aerobic sources of energy, which means they're anaerobic sources of energy. So it's done without the presence of oxygen to be involved. The last one, this fourth one, is a anaerobic respiration of ATP used for muscle contraction. And so this is done by using up, I believe it's part of the glycolysis cycle perhaps, and it's done, by lactic acid. And the mitochondria will allow you to produce more ATP within the cell. So this is done in a oxygenated environment, so aerobic environment. And I believe this will allow you to go with hours worth of ATP sources. So let's say you're standing for a long period of time, you may be teaching a class for hours or something. Most likely you're drawing a lot of energy from this aerobic source of energy. Okay. And include relative amount of each source available to the cell, the advantages and disadvantages of each type, under what conditions each might be used. Yeah, so I forgot to mention that. So the first condition, where it's just cytoplasmic ATP, this is just the initial quick ATP that's needed. You can say maybe your eyes to be able to move different directions, just for a split second, will probably use up this cytoplasmic ATP. And then the M line one is maybe if you're holding a pose or clenching your fist to hold a drink for a couple of seconds, maybe uses that. And then the glycolysis one is a good example of maybe a quick sprint. You can go from the couch to the fridge just really quickly. Okay, so there's two main types of smooth muscle. You have, you have a tonic smooth muscle and phasic smooth muscles. Now, each ones are different due to its how the cycle of the myosin binding the actin relatively works. Phasic will have a step where it can actually hold its, it can skip this initial step to be able to continue its cycle of contraction. Typically, this will involve MLCP, MLCK being placed in specific places. MLCP will stop the cycle, bringing it to a relaxed state, but the introduction of MLCK will allow you to skip a step because of an extra phosphate. And then the tonic smooth muscle cycle involves a really slow binding of ATP after its contracting state, and it'll become what we call a latch state. It'll take a while before the muscle will relax due to ATP unable to binding it really easily. And so those are the two different types of smooth muscles. Yeah, so how it starts is you have myosin and actin with its tropomyosin. Once you introduce calcium to the system, that means the troponin will bind to tropomyosin and actin, allowing those binding sites to open, and boom, myosin will now start to bind to it. It has an ADP head with a extra phosphate head, just kind of off to the side a little bit. But it would allow it to attach, and that energy requires to attach it will cause some heat or energy to be released from the between the binding sites, and this will allow that ADP and phosphate to bind together and become ATP. In order to release this, we would have to have the phosphate and the ADP to fall off of the cell, or sorry, the myosin, and this will unbind, or actually this will still leave the myosin binded, but won't be able to have the energy to release it from actin. In order to do this, we introduce ATP again, and this will cause another energy source or heat to be released. And this heat also pulls the myosin head away from the actin, which will then allow tropomyosin to come back and cover the binding sites of actin. And hydrolysis will turn this ATP head back to its first initial ADP head with a phosphate group. So that is a regular skeletal muscle system. Now it's different with tonic and phasic smooth muscles. The only difference is introducing MLCK, MLCP in specific places. Mainly, I guess, MLCP. So the MLCP is different in the later stages of tonic compared to it being introduced a little earlier in phasic, which allows ATP to bind and possibly skip a step if it wanted to allow the muscle cell to keep contracting. Okay. So the difference between smooth muscle and skeletal muscle, structurally, I believe it contains different thresholds of for the action potential, and it also contains different proteins, such as the tropomyosin doesn't actually exist, or actually I think it's troponin. Troponin doesn't exist in smooth muscles, but it does for skeletal muscles. Functionally, skeletal muscles, we leave it voluntary. It may fatigue really quickly, but overall it's really powerful, it's really strong, it gets a lot of blood flow to it. As well as smooth muscles, it is an involuntary movement and contraction. Luckily, we don't have to think about how to keep our heart pumping. And so it doesn't really fatigue really well, actually. It's pretty good about running itself, keeping it going forever, and hopefully it doesn't tire out, right? But it does a really good job at not doing so. So functionally, that's the case. Regulation of those muscles, I think skeletal muscles are, obviously voluntary. We can move these at will whenever you want, but it's also kind of reflexive. But yeah. Okay, and we want to describe and explain and then compare and contrast muscle sensory receptors. So within a muscle bundle, we have what we call a spindle apparatus. The spindle apparatus is filled with two things. They're sensory receptors, and there's two types of sensory receptors. You have a chain fiber and you have a bag fiber. Now, the chain fiber's role in sensory reception is to tell you the duration of a contraction, and the bag fiber is going to tell you the magnitude or the strength of a contraction, how tightly you're contracting a specific muscle. These allow you to feel and like understand like where your muscles are in a space in your environment and how much force is being used too. We also have uh, a tension sensory organ called the Golgi tendon organs. This is kind of like right here above or around the patella. It's that thing where the doctors, when they hit your knee and your leg flexes up, that's what it is. The tension felt from the hammer hitting your knee pulls or creates a tension on your quadricep, and its natural reflex is to contract it to keep it from stretching too far. And so thus, that's why your knee kicks your foot up whenever it's hit with a hammer. So this stretch reflex is wanting to do that. And then it sends a receptor or like a sensory neuron to the dorsal part of your spinal cord. That's just where sensory neurons typically enter. But this Golgi tendon neuron will also go through there. What it will try to do is activate an EPSP or an excitatory response to your quadriceps that's on the front that's being stretched, and instead it will try to contract it. And then at the same time, this Golgi tendon is going to send a IPSP or a inhibitory receptor to the opposite muscle that does the opposite function, which is your hamstrings. This is going to relax your hamstring so that it was able to be stretched and the quads to contract and pull their lower leg forward. And so the withdrawal reflex of that is to come back for, it's the opposite of the stretch reflex. Okay, that is everything. Thank you for listening to this. This was Exam 2 Essay 1 video for Bio461. Thank you for listening.